Hi, everyone. I'm Gela Socarras. I'm a postdoc here at the Broad, and I have the pleasure of just introducing our speaker for today's seminar series. Um, Today, the person that's going to be presenting is Dr. Gómez-Inclán. She is an early career researcher with a lot of experience in culture editing and differentiation of human pluripotent stem cells. Her career has focused on a lot of things, including modeling interactions to dissect their pathology, aiming to aid in their prevention or treatment. And during her doctorate over at the University of Queensland, she generated brain and liver cultures, both 2D and 3D, to study for rare autosomal disease and ataxia and, excuse me, telangiectasia. Currently, in her postdoctoral position at SMBS, she's using iPSCs to generate hypothalamic brain organoid models. Over the last few years, Dr. Gómez-Inclán has done a lot of work in her doctoral research, identified the post-translational modification for the catalyzed proteins that could alleviate the inflammatory response in patients with ataxia telangiectasia. In addition to all of this, she uncovered differences in the toxicity of beta amyloid peptide variants in the cortical brain organoids and focused on unraveling the diversity of physical and protein biomarkers of cellular senescence in stem cell-derived mesenchymal stem cells. A critical fate currently being investigated by a lot of different trials and for treating multiple conditions. I know this is a rather long introduction, but I cannot speak highly enough of her work. And at the current time, Dr. Gómez-Inclán is developing a robust protocol for hypothalamic organoid differentiation at SBMS, and her research is advancing frontiers of brain disease modeling. Overall, in the last 5 years, Dr. Gómez-Inclán has developed a robust and diverse skill set in the area of stem cell biology and differentiation, and she has pinpointed and addressed a lot of critical knowledge gaps across diverse research settings. I think that is quite a bit of an introduction, and I have the pleasure of presenting today's speaker. So, Dr. Gómez-Inclán, once you're ready, we can begin.
Yeah, thank you very much. Yeah, so thank you so much for having me here. I am very grateful with everyone involved in the organization, and also with Cynthia, that I think she was a contacting point for this talk to happen. So I work in Australia at the University of Queensland. Right now here is like 5 a.m. in the morning, so bear with me if my language goes to mysterious places. I work at the School of Biomedical Sciences at the with Stefan Thor. He's the leader of the group. And yeah, I'm going to be presenting you what I have been doing this past year in my first postdoctoral position with circadian rhythms and hypothalamic organoids. So I just want to give you a little bit of a background of who I am, what I have done. So pretty much I was born and raised in Mexico City. So if there is any other people from Mexico, I am a Chilanga completely. And I thought to share this beautiful map of what Mexico City used to look like when the Aztecs were around. So it's a map of Tenochtitlan. I did my bachelor's studies at the UNAM, Universidad Nacional Autónoma de México, and I was very lucky to do the basic biomedical research program. And I say I was very lucky because this program allows us from the very start of our bachelor's to be a little bit hands-on in different research groups. So I was involved in some research in the Institute of Biomedical Sciences, in the Institute of Cellular Physiology, the Faculty of Medicine, and I also did an internship in the Stowers Institute for Medical Research in Kansas City. After that, that's when I came to Australia. There is this little product called Vegemite. If you haven't tried it, I would suggest you to have a look at it. It's rather interesting. And here I started my PhD at the Australian Institute for Bioengineering and Nanotechnology. That's where I was doing a cortical organoids, but we also did liver organoids and also 2D liver and cortical models. And then I just joined like a little bit over a year ago in the School of Biomedical Sciences. So with no further ado, I will continue with talk of the actual science. So in the lab, we focus on the hypothalamus, which is a very small area of the brain. It actually accounts for around 0.54 of the total volume of the whole human brain. However, although it is very small, it is also very, very diverse. So the hypothalamus can be divided into four different regions, and then they are further subdivided into nuclei that are pretty much functional units within the hypothalamus that are involved in very important physiological processes, such as feeding behavior, thermoregulation, body fluids. So this would be more like blood pressure and things like that. Sleep-wake cycle, puberty and fertility, so things related to reproduction, also the aggressive behavior and stress response. And the way that usually the hypothalamus does all this work is through the release of neuropeptides from the hypothalamus to the anterior lobe of the pituitary gland. And then other hormones are released from this gland to other organs where it exerts their effects. So just to give you an example here of one of the pathways, for instance, with cortisol release, the corticotropin releasing hormones from the hypothalamus would be released to the anterior lobe of the pituitary gland, and then it will induce the cleavage of the opioid melanocortin hormone that will result in the generation of this peptide that is called adrenocorticotropic hormone. Those are a mouthful. And then they go to the adrenal gland, and then that's where the cortisol gets released. So I am telling you about this very complex little area of the brain that mediates many physiological processes. And anyways, the difficulties or the complexity doesn't stop there, because these processes don't happen constantly even during a day, but instead they are regulated within a day. So just to give you, staying with the example of the cortisol, what we know is that in plasma, the cortisol will vary, right? And we usually have a peak right before, in humans, right before we wake up. Then if we go and check for ACTH in plasma as well, you could notice that there is like a mild increase as well, usually a little bit before the cortisol release. And then what we know about the CRH in the cerebrospinal fluid in humans is that there is also a small peak a little bit before the other two happen. However, as in my cartoon, I think you cannot see here the hypothalamus, right? So this is not a mistake. We can actually not access this area of the brain that easily, especially in humans. Like, it's not like we can just like assess systematically this stuff. So we have used different models, like for instance here in rats, that we know that the CRH transcript in the paraventricular nucleus, where it is actually produced and then later on released from in rats, it actually also varies within the day. So these modulations are very important for our physiology, and these kind of rhythms are called circadian rhythms. And they are just called that because in Latin, circadian means about a day. So that means that these are processes that happen every 24 hours, sort of deal. And the basic molecular mechanism that regulates the circadian rhythms won a Nobel Prize in 2017, but it is pretty much in a very few words, is a feedback loop, negative feedback loop of transcription factors. So in 2004, there was this beautiful experiment, very elegant, where they fused one of these the genes of the transcription factor to luciferase in order to assess in explants of mice how does a circadian rhythm looks like. So it turns out that whenever these tissues are outside of the body, of the system, of the physiological environment in these animals, you can see that different tissues will react differently. So some of them will keep the circadian rhythm, but maybe the amplitude will change, maybe something will happen. Some other tissues will almost lose these waves. And so maybe some others will still have waves, but they are not exactly at the same time as others. So this is just telling us like the machinery of the circadian rhythms and the actual waves of the circadian rhythms pretty much happen everywhere in our body. However, they need to be synchronized, all of them, so that all the physiological processes happen as they are meant to. So how this happens is via the site givers, and I think that's pretty much German, and I have no clue of German. So apologies if I am not saying this in the correct way, but they are pretty much exogenous signals that will indicate what time of the day it is. So there are cues like light, eating, and exercise, and these cues, these signals will reach the pacemakers in the body. That is pretty much the circadian machinery that will then inform the rest of the body to be synchronized at the same pace. So I just want to stress out, this is a very complex system, and we're very interested in observing a little bit closer to the hypothalamus and how these dynamics with the neuropeptides happen. But in the hypothalamus, and you already know we're working with organelles, how do you pipette in the time? So if we think about the site givers that I was telling you about, light, food, and exercise, well, light is definitely not a goal because we don't have the machinery that it requires to detect light. Food could be, however, just like getting rid of the food of the media or not feeding the organelles could be a little bit problematic in the sense like it is also supporting all of these neuronal networks, so we are not keen to do that, and exercise is definitely a no. However, like second order site givers or a little bit of the response of the first order site givers are hormones or metabolites and changes in body core temperature. Hormones are already something that we can actually pipette in, things like melatonin, cortisol, and insulin. Particularly cortisol is interesting. Probably you know this, back in the 90s and early 2000s, there are heaps of experiments and heaps of reports using dexamethasone, that is a steroid that is fairly similar to cortisol. People have been using it in many different in vitro settings for synchronizing circadian rhythms. And in terms of metabolites like glucose and lipids, this reminded me to the usage of maybe FBS, that is like also very commonly used for circadian synchronization in vitro settings. So those two could be. Core temperature is not a good one, just because the fact of our incubators in the lab, they are usually not exactly that good at maintaining the temperature when it comes to very little changes, and that's what we would be facing. So probably nothing more than 0.5 changes in Celsius. So that is also something that we wouldn't be that keen to try. So having kind of an idea of how we would like to do that, we just want to keep in mind that 27% of the coding genes cycle in at least one gene in humans. That's kind of the last report. And in the hypothalamic neuropeptides will probably be cycling, and that's exactly what we want to focus on. So having that in mind, I just want you to come back to think with me about the model that we need to use for trying to make these studies. And so you already know we are focused on human. That's what we want to study, and that's where the power of the stem cells come, where you actually have these pluripotent stem cells that you can differentiate into different models. Something that is very important is that these cells must have the circadian machinery. I have told you that most of the cells have it, but not exactly all of them. So for instance, pluripotent stem cells don't have all of these expressed. So when you do the differentiation, you just need to make sure that you have them in your differentiated cultures. Now we want to have a system that is isolated, right? So you have seen like it's so robust systems and very complicated systems. And the hypothalamus at the end of the day works like an integration hub of a lot of different signals. And then the output is also so diverse in terms of all the physiological aspects that it can regulate. So you would have a lot of confounding variables when you are dealing in animal models, which of course you can dissect and take advantage even to some degree of them. However, we think that having a very isolated system will be very beneficial. And that is why also we think organoids are a very good idea. And of course, cellular diversity is a must for us, having in mind the diversity that the hypothalamus has. So there was in 2024 or 2023, there was this report that there are around 108 different clusters of neurons in the hypothalamus. So it's an extremely diverse area of the brain, and we would like to capture at least a little bit of that complexity. So that brings me to what people have been publishing before in terms of hypothalamic models, human hypothalamic models. And there have been, to the best of my knowledge, and probably there are maybe a couple of more now, but 18 research articles coming from 80 different research groups. And what they have been doing is exactly taking pluripotent stem cells and differentiating them towards hypothalamic regions. So as you can see, there are 2D and 3D. And something to bear in mind here is that when you're doing 2D cultures and you're differentiating them, most of your cells will be seeing roughly the same things. So you will end up with a very homogeneous, nice layer of cells. However, when you go to 3D, you have, yes, your cultures are facing still all the small molecules and everything that you are giving to them to cue towards the differentiation process. However, because they are so close together, they will start secreting even factors themselves that will help the self-patterning and will help this generate this diversity of cell types. So for instance, in the organoids, you often expect to have like some astrocytes, some oligodendrocytes as well, along with your neurons. But something that I should also mention is that as any other model, organoids are not the perfect model, and there are a couple of reasons why. So one of them is that they certainly don't have a microglia, the resident macrophages of the brain, and that is because of the protocol that we do, because we cue the differentiation towards ectoderm. So microglia is not coming from that germ layer, so we cannot get microglia. Another thing I will be talking about along this talk. But however, we know that there is people that are doing hypothalamic human organoids, and we decided to give it a go in our hands. So we just took one of the protocols, KIAN 2018, and we enriched it with some of the other protocol from Merkel 2015. And what we have is this protocol that, as I was telling you, first you cue the differentiation towards neuroectoderm. This is a very basic step with a dual SMAD inhibition that people do for cortical, but also for other brain organoids. And then you have to signal for in what region of the brain the hypothalamus is, right? So the hypothalamus is in the ventralmost part of the brain. So it often receives a lot of signal of sonic hedgehog. So that's exactly what you have to add in the media. And then for the anterior posterior axis, you need to add a proper level of wind signaling. We do that with Win3A. So that's pretty much just positioning or just cueing where in the brain you want this. And then in the next steps, pretty much I heard earlier, hypothalamus and maturation, you are just promoting this exit of the cell cycle for having your neurons, and you're promoting the maturation of the neurons. And in this protocol, we ended it at day 15, and we did that because that's essentially where this other protocol that we based it on finished. Something to have in mind is when we were comparing all the other protocols that I was showing you before, the more small molecules, the more cues you add to your differentiation protocol, the shorter it seems to be the time that you have to wait to have an end result, right? Because you are kind of accelerating the developmental process. And there are protocols for even hypothalamic models that they add much less small molecules. However, they need to wait like around 200 days to have an organoid, which might be fantastic. However, we wanted to have a little bit of a shorter protocol that we could play a little bit quicker. So this is how our organoids look like. And we decided to do single-cell RNA-seq just to analyze what we are actually getting from there, and using some data from already published articles, we could see that our organoids were at least fairly similar to both adult hypothalamus and embryonic hypothalamus. And when we have a closer look to our single-cell data, so we are happy to see that we do have neurons, we have like neuroblasts, tanocytes, so that's all good, but we also have a lot of radial glia. That means that we still have a lot of progenitors going on in there. So this seems that our day 50 organoids are a little bit early in development, and that brings me to the second main concern when you are using organoids in research, and it's because they often resemble embryonic stages, just because the stem cells are pretty much resemble very early stages. So something that I can tell you already is that if we leave these organoids to day 80, something like that, then we start seeing markers for astrocytes and oligodendrocytes. And that is very good, and that is exactly what we want to have. And they often appear later in the development, very close to the postnatal or the parturition time. So at least we have those markers now. So we are confident that we have organoids that are good enough to keep going with this, especially because we can also see the representation of 8 different nuclei of the hypothalamus, which is very reassuring. And we are especially interested, for instance, in the lateral nucleus, just because this one is very, very associated with sleep. Arcuate, that it has to do a lot with metabolism. Paraventricular, that is the one that is involved with cortisol and all the pathway with cortisol. So those 3 we are particularly interested, but to be honest, the more the merrier, right? In the ideal world, we would like to have an organoid that recapitulates the whole repertoire of cells that we see in the human brain. So as we speak and as we are doing these experiments, we are also trying to improve our protocol. However, this is a protocol that we have currently, and we are fairly happy with it, especially because we also have the expression of the circadian genes that we were talking about. And as you can see, it's pretty much a salt and pepper expression. So that probably means that our organoids, although they are expressing the machinery, they are not synchronized. So the question would be, can we actually synchronize them? And that brings us to all those things that we were thinking before of what to use. Can we do actually dexamethasone or FBS? So that's exactly what we tried. We took the protocols from many papers, like there are tens of papers doing this kind of synchronizations in other cultures. And what we could see is that upon the treatment or exposure to the dexamethasone or FBS, we could see this gene cycling. For the rest of the talk, I'm going to be focusing on the dexamethasone treatment, not in the FBS, but we were very happy to see that we can actually synchronize them. Then we decided to go ahead and start exploring what is happening with these organoids over time via Volcarni-seq. And what we can see here, I'm just showing you 6 different neuropeptides. So for instance, here the first one, this one is hypocretin. This one is very associated to sleep. And what we can see is like this cycle, and we actually didn't know how it would happen because like this one is known to be upregulated when we are awake. So that is interesting to see that just modulating the circadian machinery with dexamethasone, we can see this increase. AVP is a favorite of mine just because this one is vasopressin. This one is the one that is regulating blood pressure. And as probably you are aware, problems with blood pressure are very common worldwide, but particularly in our Latin communities, that's a big, big public health issue. Then we have CRH, the one that we were talking about, so the corticotropin releasing hormone that is upstream from the release of cortisol. And so we were not expecting a dynamic like up, down. So we were expecting more like a dynamic of this sort. So we are very happy to see that it works almost like a little bit of a control that our system is probably actually working. Then GnRH is gonadotropin releasing hormone. This one is associated, of course, with reproduction and fertility. And KIS1 is associated with the onset of puberty. So these two guys are pretty much associated in reproduction, but we're still not touching that yet. But that's something very interesting that actually one of our PhD students is kind of going deep into this. And the last one that I want to show you is PMCH, because this one is also associated with sleep, and it is also meant to be increased when one is awake. And interestingly, like we can see at least at 36 hours, it seems that it is up both in hypocretin and PMCH. So for this talk, like I want to share with you like our surprise, like do we have in our hands something that would mimic a sleep model in hypothalamus? That would be, we would be very happy with it, to be honest. But this is a very complex physiological process, so we want to try to figure out whether this could work as a model or not. So just to give you a little bit of more insight about hypocretin, so this is exactly how this looks like, and this is in mice. So it increases the number of neurons that express hypocretin. We don't know which are the cells that usually don't express it and then express it. We have no clue, but we know that there are more cells that express it, and that per cell, there is more intensity of hypocretin also, or more content and more transcription in each of those cells. Hypocretin is a gene that upon translation, it can get cleaved into two neuropeptides. One is called orexin A, the other one is called orexin B. So for the next slide, we tested orexin A via immunofluorescence. And what we can see is that over the time in our organoids exposed to dexamethasone, we can see an increase of orexin A at T12 and at T36, and this increase is very evident. And here is just the quantification of this signal, and we can see that it follows the same trajectory that the transcript does. This is the same graph that you saw in the slide before. We are also doing, and this is preliminary data, we are doing some ELISA tests where we can see that by ELISA, at least we can detect that 36-hour peak. So again, suggesting like maybe that 36-hour peak would be very interesting if we want to try to focus a little bit on sleep. So again, because this is a very complex process, physiological process, we want to try to figure out other ways where we can explain it. So this could be a cellular senescence. Maybe you are familiar with this. Like you have to gather evidence of like many different experiments to say like, okay, this is cellular senescence. I think this is what we would need to say like, we have a model of sleep. We would need a lot of different experiments and a lot of different hints that would suggest that. And because of that, I found this paper from 2012 where they were pretty much trying to figure out the transcriptomic signature of what being awake would look like in vitro. So the way that they did this was pretty much they used cortex of mouse with sleep-deprived control and recovery. And then for the in vitro setting, they cultured the cortical neurons and they added a cocktail that pretty much activate synapses or just synaptic activity. And they call that this kind of active awake neurons. And this is the list of genes that in theory they should be up in vitro, as well as they see this in the cortex. And then there is like the list of genes that should be downregulated. Please bear in mind this is mouse, not human, and this is cortical, not hypothalamus. So this is just some of the terms that I'm trying to come up to figure out whether this is sleep or not. However, interestingly, for the set of genes that are actually or should be upregulated when the cells are awake, that means at 36 hours for us potentially, I found these genes being a little bit higher in expression. Exactly what this means, I'm still not sure, but we are going further into this. And interestingly, I found like this set of genes that are actually upregulated at 48 hours, and there are a few of them. So I'm not exactly sure what this means yet, but it might be that this is just like a little bit of a late signal that these were awake or that they are awake. Because we don't really understand the dynamics of this anyways yet. For the ones that are downregulated or that should be downregulated, I found only these three, and it is not that evident, but we are going to continue to check this further. So what other things can I do to try to figure out this? So of course, we need to do immunos also for PMCH, for instance. Also, MPVF is a good candidate. MPVF should be potentially downregulated during awake stage. We want to keep doing transcriptomics, just the analysis of it. I am a wet lab and I am transitioning a little bit to analyze this in a little bit of more, computational area. So using a little bit of R and Python, and for sure, I don't know if you guys, but I find that challenging coming from only a wet lab experience. We also want to analyze the secretome via mass spectrometry, just to have like the full breadth of neuropeptides that we can detect coming out from those organoids. So so far we can detect via ELISA, the ones that we have tried, that are CRH and orexin A. Those are the ones that we have tried, but we would like to have like the full breadth. So we have some samples down in Melbourne. So this is happening as we speak. So fingers crossed we will have a lot of data coming from this experiment. We also would like to check neuronal activity via multi-electrode arrays. The only thing is that often when we think about sleep, we think about these waves of sleep. Problem is, those waves are coming from the cortex, not from the hypothalamus. And apparently the hypothalamus is a rather quiet space. So I am not sure what we would be looking at, but any information would be good information. So this is probably anyways a very valuable information. And also another thing that we should do is check for cellular metabolism. So we have seahorse, so we're going to check for oxygen consumption, but also we would like to check for maybe a mitochondrial activity because it has been seen that it is usually upregulated when one is awake compared to when we are sleeping. We have some other future directions. So for instance, how much of this is mediated by synaptic activity. So we are already doing some experiments with some inhibitors in there just to try to figure that out. What is the effect of different site givers on different cell types? So we are currently doing those experiments with melatonin, and we are going to be starting with insulin. Is there a hierarchy in the response of the hypothalamus? Because the main contribution or the major site giver would be light, right? And light will directly go to the hypothalamus and regulate there, but it also is receiving these cortisol signals, and we can very well see that it is responsive to cortisol very evidently. So what kind of hierarchies are there? Or maybe there are not hierarchies and they are just integration of signals, but how they happen, we don't really know. Are there differences in males and females? In the hypothalamus, there are differences. For instance, we women have more KIS1 neurons. Remember, KIS1 is a peptide that I told you that is involved with puberty. So we have more of those neurons compared to males. What happens with different genetic backgrounds? Or even in disease, like what happens with narcolepsy? Like for instance, in narcolepsy, often or a lot of the patients have mutations in the hypocretin gene. Because of my background and because of how the climate, the biological climate that we have is what happens in other cultures. Like what if we do organoids of different parts of the body, like how comparable would be the responses to these site givers? And this is even longer term, but we would be very interested to see how this plays out, because for sure there's going to happen. Like, how would it look in a body on a chip? So that means, like, what if you put organoids of different areas of the body and you connect them, all of them? And then what if you set time in the different ones and see how this whole system reacts? So pretty much just dissecting every single way of this machinery. And that is what I think would be happening in the future, like, I don't know, like 5 to 10 years, hopefully, and hopefully we're there to see that happening or make it happening. So without further ado, I want to thank you all for your attention. Of course, I want to thank my boss, Stefan, and Ashvati, that she's my partner in crime in a lot of these things that I have been showing you today. And I'm ready to take questions.
Thank you very much for the presentation. I just want to mention that there's going to be a short survey, and if you can please fill it out, it'd be greatly appreciated. First question is from Daniel Gonzalez. A very interesting model. Maybe I missed it, but how do you reconcile the lack of sensory machinery with the circadian changes observed in the organoids? Do you think the organoids behave like this regardless of their environmental cues?
Okay. So we are actually running the controls where we are having our organoids without any cortisol during the whole span of the experiment. So we are taking organoids every 12 hours as well, just to make sure that the baseline doesn't cycle. However, the data of the single cell RNA-seq suggests that they are all that they are not synchronized. So that shouldn't be an issue, but let me know if that is what you meant and if I actually answered your question. Maybe the other aspect of your question that, because I did say that there are some cells that do not have the machinery, but we can certainly tell that our organoids do have the machinery, so that should be up and running. I hope that that answered.
Okay, any other questions? It was a very interesting talk.
Sure, I'll throw a question in. For the secretome, it's amazing that you're considering that as a future direction. Are there other things you want to look into in the secretome beyond proteins? Like, are you thinking perhaps that there are different things being secreted out from the cells?
Yeah, so with the secretome, what we are doing is that we are depolarizing the organoids just to make sure that they release all the goodies that they have in there about to be released. And what we would like to see is pretty much the whole landscape of the neuropeptides that would be released at a given time. And that is because we are very interested in the metabolic aspects of it and like just a very physiological output of it. So it's such a diverse group of cells in there that impact many things that are actually associated to many problems that we have right now as major public health concerns worldwide, that we want just to try to make sure that we pay attention or that we can see as much as we can in terms of all these neuropeptides that can be released in our model.
Fantastic, thank you.
How many different environmental stimuli have you applied to the models to see if it disrupts the circadian rhythm?
At the minute 3, so dexamethasone, FBS, and melatonin. And we are using melatonin as well because it's also pretty much physiological, right? I mean, it is produced in the pineal gland, so it's not exactly coming from the hypothalamus, so we can deliver it extrinsically to some extent. So that is why we went with that. So although it's an artificial setting, we're trying to mimic as good as we can the physiology.
Okay. We do have another question in the chat. For the type of model that is used, how consistent would it be to measure the markers from rat and organoids? I guess rat and human organoids.
For the type of model, can you repeat the question?
I think there's something missing, but the question is, for the type of model that is used, how consistent would it be to measure the markers from rat and organoids?
Okay, so maybe the comparison between models. You know what? Probably fairly similar, but we have to pay attention to something very basic. Rats, for instance, are nocturnal and we are diurnal. And that difference happens in the hypothalamus. So just for the sake of studying sleep, the things will be like the other way around. And where the connection becomes different between nocturnal and diurnal, it seems to be coming from the innervation of the suprachiasmatic nucleus, that is one of the nucleus of the hypothalamus, to the paraventricular nucleus where the actual CRH is actually released from. But we're not entirely sure how that machinery works. So that's why we have such a big issue at the minute in that level of complexity. When you want to go more granular, that's where using other animal models, it might be a little bit tricky to make the full comparison. However, for sure, like all the animal models are very valuable and will continue to be super valuable. But you just need to think about what exactly you're asking and whether it actually makes sense to compare it in that particular, to answer that particular question. That's it.
It's very true. And I mean, keeping those details in mind is really important before we come to a conclusion.
Yeah.
Yes. Okay. I'm going to give people maybe a few more minutes to drop questions. Speaking of models, you said you wanted to compare male versus female biology when it comes to hypothalamus. I think there was a paper circulating a little while ago saying that there is a distinct difference in terms of the hours of sleep or how much rest is needed. How do you think that's going to impact potentially the findings that you get?
That's a very interesting question. I am not too sure. And the thing is that males versus females is more complex than it looks like, because it doesn't only come with just the chromosomes that you have, but during development, for instance, we have waves of estrogen and testosterone flowing that apparently regulate to some extent the identity and the function of the hypothalamus for later on. So at this stage, what we are doing is first like trying to figure out, do we have the proper model for analyzing males versus females that is more physiologically relevant, that it is not only having the correct chromosomes. So that is something very important, particularly for the hypothalamus, because it is involved with all these reproduction systems, right? How would it play out later on? Say we have a good enough model that we consider that we want to move forward with it and just check up sleep. I'm not too sure. I'm thinking it's going to be very different. Like, I hate to say this, but my partner, like, he eats like crazy and he doesn't gain weight. But anyways, I'm always very hungry, and he's not that like that kind of things. Like we often see in just like the people that we have around. They a lot of that is happening from the hypothalamus, like all this hunger, this like how much intake, but how much weight you gain, and so it's all wired in there. I'm not saying that it's the only contribution, but it's definitely in there. So for sure, we're going to see differences in there. Also, another thing is that after menopause, there are heaps of metabolic changes happening for us. So how to model that? That's another question. And once you can model it, I'm not too sure what we're going to see. But for instance, like when women hit menopause, the incidence, for instance, for strokes go so high, just suddenly. So I'm not too sure. And that might be related to blood pressure, and that might be related to vasopressin, that, by the way, is actually released from the hypothalamus. So there are many different aspects, but first you need to kind of tune down what is the model that you are going to use. And that's what we are trying to set up first, like just a reliable model that is as physiologically relevant as we can make. And then make these what seems very basic, simple questions that actually they are not that simple.
It's true.
I had a question as well. Do you plan on inducing some of the genes associated with psychiatric disorders? Because there's a dysregulation in circadian rhythm sometimes with psychiatric disorders like schizophrenia and bipolar.
No, not at the minute. What I am trying to do via Viper, so and with CRAB is try to modulate genes associated with actually obesity. So FOXP2, SIM1, those are things that are happening in the hypothalamus that we would potentially be interested to do. Things that are psychiatric, not at the minute. I'm aware that there are a fair bit of things happening in terms of autism, and like some papers that have come like out with things like that, especially with epigenetic modulators. But we are not addressing that at the minute. But why? Like, do you work with that? Is that something that we should actually be looking at?
Yeah, so it's with psychiatric disorders, it's anecdotal, but like psychiatric patients, particularly bipolar and schizophrenia, they do have like a circadian disruption and their timing clocks are off, right? So we don't know, like from a genetic standpoint, like
so my colleague Ashwati, she's working with Parkinson's disease, and pretty much a lot of the neurodegeneration diseases that we have that are major concerns are also having a circadian dysregulation. So it seems like it is a very sensitive to some degree system, although it is very robust because people are still alive and whatnot, but it seems like it is fairly easy to dysregulate it. Same like even like with addiction, like what happens in terms of the circadian rhythms in there. It seems like it is not it is modulated somewhere in there. So that is something else to have in mind. That pretty much is regulated.
Yeah, so the thing with psychiatric disorders, it's always like a chicken or egg scenario. Like, are they having psychiatric disorders and their behavior is also dysregulating their circadian rhythm, or is it the circadian rhythm is dysregulated and inducing these things, right?
And having an isolated system then helps you out to figure out which is the chicken or the egg, right?
Exactly.
So that is why these kind of systems are relevant for doing these very basic essays here and there. So yeah, I didn't know, but that is something to keep in mind. Thank you.
And are you planning on a longitudinal long-term study and keeping your organoids as long as you can to see how dysregulated time and age causes changes?
1000%, 1000%. So for instance, in my previous lab, we were leaving cortical organs over a year to have a good model for what we would like call aging. So that is something that absolutely I have some organoids in the incubator that I'm just like keeping there forever, and then and I'm just going to have a look at them later on. But yeah, that is very, very interesting as well. So what is happening to us at the minute with this model that we just set up is that there are too many interesting questions that we can tackle. So we are just trying to span as much as we can strategically and feasibly do. But yeah, and we are of course open to collaboration. So if anyone here is like, oh, we should really do this, and let's write a grant about it, we would probably be keen.
Yeah. So my lab is working on PWS, and one of the target tissues is the hypothalamus. And,
oh, there you go.
Yeah.
That's very cool. That's very, very nice. Maybe we should talk later.
Yeah. So the manager of that, who does all the wet lab work, was here. So he might be in contact. So I'll follow up with him.
Oh, fantastic. Yeah.
All right. Do we have any more questions or anyone else wants to chime in? I just wanted to thank Cecilia again for waking up so early and doing this talk and being so bright and perky at the same time. If not, we can call it in like a minute, and you have 1 or 2 minutes to put in your questions.
Yeah, I think that was perfect. And it sounds to me like the best thing that you can go to a seminar has happened, which is new ideas for research and potential collaboration. So it's fantastic all around.
Awesome. Thank you so much for having me, guys.